morning. Today we are going to talk on physiology of food mineralization and the theories of mineralization. So we know physiological mineralization happens in hard tissues. And what are the hard tissues in the body? It is the bone, cementum, dentin and enamel. Except enamel, all other cells have specialized connective tissue and collagen, principally type 1, which plays a role in determining their structure. The cell production of organic matrix capable of accepting mineral plus the activity of alkaline phosphatase and a good supply is a prerequisite for mineralization. So when we see the formative cells, we see the we have fibroblasts, odontoblasts in the teeth, ameloblasts for enamel, cementoblasts and osteoblasts. They all possess abundant mitochondria, Golgi apparatus, rough cytoplasmic reticulum and transport vesicles and secretory vesicles. So we can see the cell is made up of all the abundant organelles in the inside. And when we talk about organic matrix, what do these cells do and what is organic matrix? Organic matrix is the base. So it can be made up of either collagenous proteins or non-collagenous proteins. So they are only proteins. And collagenous proteins, there are different types of collagens like type 1 to type 5. And what is a collagen? Collagen is nothing but the amino acids like glycine, proline, hydroxyproline. So they form chains. These chains, they form helices like a triple helix, and these triple helix forms the collagen fibers and they lay out to form the collagen matrix. So there are different types of collagen. We have the type 1 which is in the bone, type 2 is in cartilage, type 3 in blood vessels, type 4 for the basement membrane and type 5 for the placental membrane. So these are the types of collagen which is present in the body. In the bone, for bone mineralization, type 1 collagen is involved. So type 1 collagen acts as a scaffold that accumulates the minerals in the holes and pores of the fibrils. We know that collagen forms fibril network. In that the minerals gets deposited. And there are non-collagenous proteins like proteoglycans, phospholipids and phosphoproteins. The difference between the collagenous and non-collagenous is non-collagenous proteins don't form triple helix, whereas collagens form triple helix to form the fibrils. And they are not the main component of the extracellular matrix, whereas collagenous proteins are the main components of ACM. So the non-collagenous proteins are involved in mineralization of enamel, whereas in other hard tissue collagen, type 2 collagen plays a type 1 collagen plays an important role. So when we talk about enamel, so we have non-collagenous proteins which play an important role. So all hard tissues, regardless of their composition, are capable of accepting minerals in the form of hydroxyapatite crystals. So whatever the collagen type is, the hydroxyapatite crystals will go and settle there to form the minerals. What is the organic matrix of enamel? So it consists of distinct families of enamel proteins, mainly 90% of amylogenes, which contains proline, histidine, glutamine, which helps to maintain the space between the crystals, and 10% of non-amylogenes like ductilin, enamelin, and amylin, and they help in nucleation and growth of crystals. So we see here there is nucleation and growth of crystals in the enamel. So when we see the theories of mineralization, we will come to know what kind of mechanism it happens in the enamel for mineralization. So we can see here the nucleation which happens, and the enamel hydroxyapatite is deposited in the form of, when we see a section, we see a keyhole pattern during the mineralization. And what are the minerals? Only mineral which we see here is the calcium phosphate, which is hydroxyapatite. The inorganic component of mineralized tissue consists mainly of calcium hydroxyapatite, which is a biological apatite. So it has this calcium, phosphate and the hydroxyl groups. This is how the chemical structure looks like. So what happens in the enamel and dentin during mineralization? So enamel, the formative cell is ameloblast, and the organic matrix are amelogenin or non-amelogenin, and hydroxyapatite is more here, 90%, and non-amelogenin protein help in nucleation. So what are the amelogenin and non-amelogenin doing? We know that amelogenin forms the organic matrix and non-amelogenin helps in the nucleation. And we see the ameloblasts which deposit the organic matrix first, and then the nucleation causes the hydroxyapatite being deposited and the mineralization happens. So this is how an ameloblast with the Tom's process they start depositing the organic matrix on which the minerals are deposited to form the enamel. In dentin, the formative cell is the odontoblast. The organic matrix are the collagen and non-collagenous proteins. Hydroxyapatite is 67%, and nucleation takes place in the matrix vesicles. Here it is, primarily it is nucleation in the non-aminogenic proteins. Here the matrix vesicles play an important role in the mineralizations. And this is how an odontoblast will look like, which has odontoblastic processes, and the collagen is deposited on which the minerals are getting formed. So although the tissue fluid is supersaturated with calcium and phosphate ions, spontaneous precipitation of calcium and phosphate does not take place because there are inhibitory molecules which are macromolecules. They inhibit the crystal formation. These are pyrophosphate, the diphosphonates, or adenosine triphosphate. They delay or prevent the transformation of amorphous calcium phosphate to hydroxyapatite. This is the reason we don't develop any pathological mineralization in soft tissues because of these macromolecules. And there are citrate, magnesium and proteins like albumin which are also acting as the inhibitors. The components of bone matrix are proteoglycans. So what happens? First, there is an unstable initial cluster of ions which need to form a lattice structure. Further, the formation of cluster of ions requires the expenditure of energy, and an energy barrier must be overcome for crystallization. So they should overcome the energy barrier for crystallization. The deposition of mineral salts in and around the organic matrix makes it a calcified structure. There are two mechanisms of mineralization. One is the booster mechanism and another one is the seeding mechanism. In the booster mechanism, there is a concentration or action of enzymes is dependent. The concentration of calcium and phosphate ions increases to such a level that would lead to their precipitation. This is booster mechanism. In seeding mechanism, there is a presence of seeding or nucleating substance which acts as a template on which the crystals are deposited. So the mechanism of mineralization are only these two, whether the concentration gradient or there should be a nucleation site. Now when we come to the theories of mineralization, the first theory which was proposed by Robbins was the Robbins phosphate theory. So what does it say? The enzyme alkaline phosphatase present in the organic matrix of calcifying tissues hydrolyzes the organic phosphates, leading to an increase in a local inorganic phosphate concentration. In the last seminar, we talked about PPA, the pyrophosphates, which are inhibitors of mineralization, which is acted upon by alkaline phosphatase to cleave it into the organic phosphates. Then only the concentration of phosphate will increase and calcium phosphate will form and get deposited. So he suggested that this alkaline phosphatase plays a major role in the mineralization. So this elevated phosphate level facilitates the formation of hydroxyapatite crystals, initiating mineral deposition. But there were objections for this theory, where it says the alkaline phosphatase is seen in other tissues which do not calcify. Say they found the alkaline phosphatases in other tissues, but there the calcification doesn't happen. So this theory is contradicted here. And another thing is organic phosphate is not sufficient to produce inorganic phosphate to initiate calcification processes. So these are the objections for this theory. And later there was another theory which is the nucleation theory or the seeding theory by Enamel in 1953, which stated that the collagen fibers in the organic matrix serve as a template for mineral deposition. And the periodic gaps within the collagen fibrils act as a nucleation sites where the hydroxyapatite crystals can form and grow. So that it can be a homogeneous nucleation or a heterogeneous nucleation. Homogeneous nucleation says that if there are any local increase in concentration of inorganic ions permits a sufficient number of ionic clusters of crystallite to form. Whereas the heterogeneous nucleation shows the presence of nucleating substance which allow crystal formation to occur in the absence of a locally increased ionic concentration. So the concentration differences also makes a different nucleation pattern. So this process is further regulated by non-collagenous proteins that modulate crystal growth and orientation, ensuring organized mineralization within the matrix. And this theory also has some objections where it fails to explain the mineralization in enamel and cartilage. So these two areas of hard tissue mineralization was not explained in this theory. And it failed to explain the mineralization in soft tissues, though it contains collagen. So soft tissues also contains collagens, But this theory did not explain how the soft tissue mineralization happens, and because collagen and soft tissues are densely packed, which impede the phosphate ion access to nucleation sites. And the third theory is the matrix recycle theory, which is the most accepted theory. The matrix vesicles, already we saw in the last seminar, is a membrane-bound extracellular structure released by the mineralizing cells like osteoblasts and chondrocytes, which initiate mineralization. And these vesicles concentrate calcium and phosphate ions, creating a microenvironment conducive to the nucleation of hydroxyapatite. We saw that it starts from the vesicle, it goes outside into the extracellular matrix. And the once initial crystals form within the vesicles, they rupture, releasing the crystals into the extracellular matrix, which they propagate and contribute to the tissue mineralization. So the matrix recycle theory helps in explaining most of the mineralization processes. In the dentin, the matrix recycle theory is the main process of mineralization, whereas in the enamel, the nucleation in the non-equivalent nucleates helps in mineralization. So mineralization in humans is a biologically intricate and tightly regulated process involving coordinated actions of cells, enzymes, extracellular matrix proteins, and ion transport systems. While several theories have been proposed to explain this process, none comprehensively accounts for all the aspects. So current understanding supports a multi-mechanistic model that integrates various elements from these theories, reflecting the complexity and diversity of mineralized tissue formation in the human body. Right?